I'm thrilled to welcome you to our precision medicine track. And I have to give kudos to my co-chair of this track, Aaron Pierce. As many of you know, Aaron Pierce was my long-term NP who got interested in research and took the Vail course on protocol writing and started writing protocols and started being a phase 1 PI in her own right. And lo and behold, she started serving on national committees, and Honor Health out of Phoenix, Arizona said, my gosh, you're operating at a really high level. Why don't you come to our center and write protocols all day long, and we'll pay you a whole lot more than Dr. Matrana does. And said, you know what, Aaron, you have got to spread your wings and fly. We're so proud of you and what you've done. But she was instrumental in putting together this track. However, she had to fly out a little early from the meeting, and so she couldn't be here this morning. I hope you guys got to see her previously yesterday or the day before she was here. But anyway, I wanted to give some kudos to Aaron for helping me put this track together. We have what I think is a phenomenal lineup for you today, and it begins with Dr. Mary Nordberg. Dr. Nordberg is a genetics expert and a genetic educator at Willis-Knighton and at Lake Charles Memorial. As many of you know, CAGLA has been a little bit New Orleans-centric in the last few years, but this year I think we've really broken out, and we have a lot of folks from all over the state, a lot of physicians and others who are joining us from Shreveport, from Lake Charles, from Monroe, and many other parts of the state. And Dr. Nordberg really helped to open that up for us in many ways. In addition to her clinical work at Willis-Knighton and Lake Charles Memorial, she is also a professor of medicine at LSU Shreveport, and she runs a genetics and precision medicine program out there in North Louisiana, and also has a regular genetics clinic there as well. She was the previous director of molecular pathology at Delta Path for years, and brings to us today just a wealth of precision medicine and genomics knowledge. So it is my profound honor to welcome Dr. Mary Nordberg to the stage. Thank you.
So as Dr. Matrana said, first of all, I really appreciate you having me here. Aaron, and we've had many conference calls, and I appreciate the opportunity. As Dr. Matrana mentioned, we don't have a big showing from North Louisiana, but I do have my one Willis-Knighton fan who has come. The other two decided not to come. So for whatever reason. And then I do a clinic at Lake Charles Memorial, and I know Dr. Bergeron was here. I saw him yesterday. I don't know if he's still here. So I work with all these people, but in full disclosure, I am not a treating oncologist. I am a cytogeneticist and molecular geneticist by training, have been lab director for genetics labs for years. In 2018, though, and for reasons I'll talk a little bit about, and Dr. Ellis talked about it yesterday, you know, technology moves pretty fast these days. So in the old days, I did cytogenetics. We had a 35 millimeter film camera on our microscope. We took pictures of the chromosomes. We went in the dark room, in the dark, developed them, printed them out, put double stick tape on the back, cut them out, and karyotype. And then digital karyotyping came along, and it's hallelujah, that's good. Then FISH came along, and it's like, oh my gosh. And southern blots are gone, automation came to molecular. So stuff moves fast. So what I'm talking about today, I have 140 cases to cover. Just kidding, just kidding. But I do have 140 cases that I've gone through, and there's a couple takeaways before I start that I would like to kind of mention. First of all, so I do genetics. So the stuff I'm going to talk today about makes germline genetics look like a cakewalk, because the germline genetics is pretty straightforward for the most part. All this stuff and all the vendors and industry out there is where it gets very complicated. I'm not going to talk about germline today because Dr. Schutz in the other schedule is talking about that in GI. Very interesting. I learned a lot from him the other night. But this started out with a thing with Lacey Vasilakis, who is a nurse practitioner at Lake Charles. And we kind of talked about we'd go to tumor board, and they have an all-comers tumor board every Tuesday. And everybody would sit around, and Dr. Fredericks, who runs the tumor board, is very good. And every time at tumor board, he writes down, we know on all these patients presented, who's getting what vendor we're using, et cetera. So kind of everybody knows. At Willis-Knighton, it's a little different. We have multiple tumor boards. So since Dr. Gallagher is here today, I hope one of the take-homes away, there's a couple things. One is integration. I mean, integration, we do not have integration with a lot of the testing labs, and I'll call everybody out in a second, but I will say integration and the ability to access these reports is key. So as I mentioned, I have gone through 140 reports. We have a new EMR. So our new EMR is Meditech Expanse. We do not have a single place for genetics. So for the most part, I have to read every single report. And the shout out I'm going to give to all the vendors. So Karis, Dr. Tyler Maddox, Kim, Gavin have helped me a lot. Dr. O'Grady and Savannah De La Rosa from Tempest have helped me. All the garden people, Tamara and Harville especially, kind of helped me pull cases, because when it gets to pulling cases, we don't have a one universal system to pull. And I think if there's any takeaway from here, for those of you that order testing or do testing, you know, Foundation has helped me a lot. I work with everybody closely. I have no disclosures, absolutely zero disclosures, but because my informal title at Willis Knighton, in addition to doing the genetics and facilitating the genetics, is sort of the unofficial biomarker navigator. And a lot of people do very well on their own, but then you get in the confusing space. And that's where I think that we can all improve. And I've heard multiple very smart people this weekend talk, and that's kind of the one takeaway is integrate if you can, and then education, physician education also, as well as patient education. Like, and we'll talk, why are you ordering the test, you know? And then if you don't like the result, you're gonna do something else. And then that's where it kind of gets confusing. So we'll do, y'all, everybody in this room kind of knows this on the overview, but we are going to talk a little bit about the terminology that gets flipped around. And some of it's used appropriately, some of it's not used appropriately, but when we talk about it, you know, the overall goal is really to help our cancer patients. I'm a huge patient advocate. I'm a past president of our Cancer Foundation League in Shreveport. We have a vendor fair, and many of you guys who work our vendor fair support that. All that money, 100% goes to our foundation. It's all about helping patients either through their treatment, through their diagnosis, or, you know, for long-term treatment selection and prognosis. And this has gotten very complicated, and like I said, I could go over a lot of cases, but I'm not. We're just gonna do a 35,000-foot overview, and then I encourage every single person in this room who utilizes, interprets, works with patients, to understand who your vendors are, who you're using, what the results are. And the results, and I'll be honest, I mean, I speak molecular, but you know, there's a lot of pages involved. And sometimes on the front page is the answer, sometimes on the last page is the answer. So you just have to be familiar. So there's all kind of different tests that we use, and everybody in the lab knows these. And they're not always 100% congruent. Sometimes they're discordant. Sometimes they're not even apples to apples or apples to oranges. And understanding that in the realm of patient is very important. And so I do spend a lot of time, in addition to germline genetics, educating patients on things like, why are we ordering Signatera? Why are we ordering Caris? Nobody talked to me about that. What does that mean? And now I have a bill. So help me decipher all this, what is happening. So the kind of confusion for people who don't do this full time is really the difference between biomarker, epigenetic, epigenomic. Had a great conversation with somebody yesterday about epigenomic changes and really what, you know, I can tell you what these 700 or 74 gene panels do, but what really is modifying those? And I know there's a molecular tumor board right after me, which is the other shout out I'm gonna give, and Dr. Gallagher can help me, is I really think having a full-on molecular tumor board is helpful, because there's a lot of information to go through, and all the treating oncologists and treating radiation oncologists and hemoncs kind of need to go over that in a section that would help. So there's NGS, and I think everybody in this room will acknowledge that some kind of comprehensive genomic profiling is absolutely standard of care for all of our cancer patients. Some more than others, some different, and then monitoring patients for minimal residual disease. But there are differences between circulating tumor DNA, cell-free DNA, and circulating tumor cells. And I hear those terms used universally, and they're not always universally. So I show this about Garden just because, one, their platinum sponsor, Tamron, helped me quite a bit on some of these. Again, I had to call on the MSLs and a lot of these vendors because we don't have a universal system for me to search and say, who are these positive? And I also say that because some of the oncologists I work with, not you, Dr. Gallagher, some of the oncologists I work with just call me and they're like, Mary, can you do a Garden? Okay, so I'm showing you this because there's 4 different gardens which require 4 different boxes, 4 different specimens type. And in our genetics clinic, we have a wall, literally a wall full of every known kit that you can imagine, germline, somatic cell, et cetera. So understand what you're testing for, understand what you're ordering, because if I draw the wrong tube, then it's going to not be accepted by the company. So we just need to be clear on that. So an easy one that we do, and this is Garden Reveal. And so this is kind of, in many ways, the easiest interpretation. And if you look and see, you know, you start out positive, and then, where's my thing here? You start out positive. This might, oh, there we go. You start out positive and you want to go negative. I mean, that's the goal. And you monitor, but here's the deal with the Reveal and some of the other tests. Do it in a systematic manner. Don't just do it once and forget about it, because it really does, it's not a static test. It's not a static test at all. It's a dynamic test. Now, some of these patients, this guy was out for 40 weeks. They tested positive. You expect that in the beginning. Negative, that's great. Everything's going well. And so treatment decisions are made on that, and you have to kind of decide globally. That's where a molecular tumor board comes in, because everybody's in the room together, everybody's talking about it, and one person may have one, and they're like, have you thought about this? And that's the other thing. So here's one, you know, you're clipping along, and on this last patient, I just want to remind just real quick. On this woman here, this is a lung cancer case, I'll just tell you. And at the end of this talk, I'm going to tell you kind of what's going on with her now. Because now she's switched from hemo, well, she's still with hemo, but to radiation oncology. And we're doing something totally different with her now. And then here's one, you know, and I've had great talks with people from Natera and some other places about sustainable negativity, transient negativity, or constant positivity. So on this person, there's a little blip here in the middle. And so basically, you have to make a decision. Some people, and I'll show you an example in a second, are watch and wait, and they do watchful waiting. Other people move on, and then they do something else. So then we start getting into, I don't like this result, so I'm going to use somebody else. And so Dr. Tyler Maddox helped me with a lot of cases. I'll show you in a second, too, from Caris. In addition to Garden. So this was a case which was great because guess what? It's concordant. So this was a case from one of our oncologists that it was a suspected lung primary, but breast couldn't be ruled out. So that's where the value of all these tests come in. And every one of them, it's not a one size fits all because every test has different attributes. And there are some people, sometimes it's physician preference. Some people like the way the reports look. You know, I had this argument a long time ago, sorry, Ramsey, with Myriad. And Myriad, everybody loved the Myriad genetics germline reports because their front page is green or red, and it was so easy to read. And they hung on to that for a long time, and they do those nice brochures. But it is about ease to read and interpretation, and who's disseminating the information. But in this case, both EGFR mutations, Garden Reveal also showed an exon 19, and then on the AI for Caris, they showed it was a 99% lung cancer adenocarcinoma. So that was great. It's great. And then the other thing is, and I know we talk about this, but as a pathology person in the lab, I have spent numerous hours looking down microscope, doing macro dissection, micro dissection, all these kind of things before everything got really complicated. And, you know, we're blowing through that tissue on IHC stains. And Dr. Ellis mentioned that in the opening thing on Friday, whatever, yesterday, I don't even know what day it is, said that basically the movement and we're not going to hang up on pathology right now, but the movement toward liquid biopsies, once we kind of understand the whole thing, I think is coming. I mean, I've seen it happen with chromosome analysis. I mean, I've done amnios, I've done chromosomes, but the whole cytogenetic thing is quickly also falling away due to technology increases. Tempus, so we use a lot of Tempus, and I knew there was a molecular tumor board, so I didn't present a lot of Tempus cases, but Dr. Grady and Savannah have helped me a lot. We do a lot of paired testing, lots. And, you know, one thing I will say, and again, I'm totally no disclosures. I'm just giving information. So with Tempus, you know, we get the tissue, we get the blood, and now they have paired with Ambry for their germline, and so we use Ambry. We do their RNA testing also. So it gives you another option, but there's a lot of options, and that's where I think as a user and a provider for treating patients with cancer, what test is best for my patients? I mean, we don't always, and again, sometimes one test does not fit all, but then when you change, you may or may not have a concordant result. So I will also give full disclosure, I use Prezi a lot, love Prezi. But when Chad told me I had to do it to PowerPoint, I moved it over to PowerPoint. And there's some little small things that my genetic counseling student said she checked everything, but note to self, I'll do it myself next time. But there are some things that didn't transfer over. I just show you that because it's just one of those things that I'm like, oh my gosh, Prezi's great. But when you transport it to a Microsoft product, it doesn't always work. So anyway, having said that, this is all tumor, you know, either tumor informed or tumor not. So this is a classic example of circulating tumor DNA. Cell-free DNA is different from circulating tumor DNA, although circulating tumor DNA can be found through the genome with the cell-free. Easiest example of cell-free DNA is NIPT for non-invasive prenatal testing. I mean, for those of you or anybody who does prenatal testing, trisomy 18, 13, 21, and all the other X and Y, I mean, that's the holy grail of NIPT. Doesn't always inform, sometimes the amnio is still necessary, but what's interesting is the labs that do amniocentesis for confirmation are few and far between in these days. So anyway, so we know that ctDNA, you know, breaks off from the tumor, can be found in the peripheral circulation, and that's really kind of what we do. So this is the holy grail. This is the holy grail of everything. So no matter what you're looking for MRD or whatever, you know, we expect that at diagnosis, you're high, okay? I mean, that would be a great target. It's great to have a target. It's like Dr. Lippman was talking yesterday about lung cancer. And, you know, if I have lung cancer, I hope to goodness I have a targeted mutation. That's what I want. I don't want to be, and I sit through a lot of tumor boards, and we have patients who come who, you know, do the Tagrisso alter because they're EGFR positive or something like that because it's been a value of their life. And so again, you go down and then early recurrence monitoring and then treatment response. So the only caveat I'm going to tell you here is this is a great graph. I don't think in general as an entity that we use it fully the way it's supposed to do. For example, do you keep treating a little bit longer, even though it goes away? Do you not treat longer? And again, those are decisions that need to be made after time as far as who does what and what the result is. So then there's tumor naive and tumor informed. Pros and cons of every one, but just be cautious when you guys are ordering tests or working with your oncologist or doing them yourself. You know, sometimes this is good. Sometimes this is good. Tumor informed is great too, because now they're mapping my tumor. But I could have 100 other cancers growing in my body, and this test would be negative as tumor, because it's working off my primary tumor, right? Different, I mean, molecularly similar assays, although the endpoints are different, but the starting point is what's different. So Signatera, we use a lot of Signatera. Dr. Petty, who's one of my colleagues at Willis-Knighton, he treats a lot of breast cancer patients. He uses a lot of Signatera. Signatera tumor informed. You start with what your tumor looks like, and then you trace it over time. And again, like that graph I showed before, you start out positive, and you hope you go negative over the course of the time. I mean, that's really what you want to do. And so the difference between comprehensive genetic profiling, which is a little bit different, and Signatera, one driver, one passenger. Drivers are different than passengers, and I don't have time to really go into all that, but they're different tests. And so if y'all are ordering the test and you're using the test, understand the differences and then be able to explain it to your patients, because they're the ones that they don't know a driver from a passenger, but they do need to know, what's going on here and how does this affect me? And then the classic decision tree is it negative? Then go with standard guidelines. Is it positive? Then you do the other things that you do and then consider clinical trials. And that's really what's going to happen. That's what's going to change the world as far as how we use this technology in general. Okay, so I have a couple of cases that we picked out that show some of the both positive ends and negative ends of this. So this is a patient of Dr. Petty, who I work with. So we always, the germline stuff, now there's tandem germline, but historically we would do germline on anybody who met the NCCN criteria for germline testing. And then the somatic cell testing or the liquid biopsies or whatever came later. But she was a high risk by oncotype, stopped endocrine therapy, no evidence of disease. But then her Signatera was positive, and so she was started on new therapy. So here she was a diagnosis, goes way up, as you would expect, and then goes down and stays down. And there's still follow-up on this patient, but we keep doing it until, you don't do it anymore. I mean, really, there is no, different organizations have different companies have different consensus on how long out you follow patients. But in general, most of our patients come with their own boxes and they're ready to have it done again. So timelines are very important. So one thing that gets me in trouble is that these aren't all my patients. So they will see one of our oncologists and then I'm asked to follow up with the cadence or whatever that we need to do. And so having a timeline, again, back to molecular tumor board, having a timeline on all these people kind of puts you on notice that, okay, April's coming up, Miss So-and-so needs to come back in and have another analysis, because that's what helps the whole process. Or we have duplicate orders. The other thing are duplicate orders within the organization that is not helpful. So here she is, low, keeps rising, therapies change, and now she comes down again. So that was a good one. Now here's the problem case. So this case is actually one of the ones that we've talked about for a long time, and both Ashley Ray at Signatera and Tamarin from Garden have helped me with this, because this is a case where you didn't like the answer and you did something else. Now, again, different testing methodologies, but having said this, she is a young woman. She has done all the right stuff. She had a bilateral mastectomy, skin sparing. She had a hysterectomy. She's had all this done. Germline testing was negative. Signatera coming up, everything's negative. PET's negative, Signatera coming up. Therapy was changed, and here she was switched, but she's still coming up. So ironically, this is my neighbor. And she asks me every time, and I walk my dogs by her house, and I have to admit that when I see her sitting outside sometimes, I just go the other way. Because we can't, and I've talked to Tamarin about this, I've talked to Ashley about this, we can't really explain this at this time. She has no evidence of disease. So Dr. Petty's approach now, and again, I'm not her treating oncologist, he's doing watchful waiting right now. We have not, now that the issue comes, here was where the issue was. So we did a reveal on her back in September, and the reveal was negative. But remember, reveal is not the same thing as Signatera, and that's where everybody kind of gets mixed up a little bit. So having said that, he has not repeated the reveal, but he needs to repeat the reveal. If you're going to do it once, do it twice. And the Signatera is still there, but no evidence of disease. Okay, so we also do a lot of KRAS, and so for KRAS, we have Tyler Maddox submitted these cases, and these are kind of all over the place. The first one is actually a cancer of unknown primary. Helped us a lot to identify that the tissue of origin was like 98% non-small cell lung cancer. Lung Assure is great. because one of our other colleagues, Dr. Master, uses a lot of Assure. Not only did he find an incidental MUTYH mutation, but there was incidental CHIP mutation. And I don't have time to go into CHIP, but CHIP is a big deal now. And then I work with Dr. Justin Black. She does a lot of, she has a clinical trial organization called Trials 365, and she's a GYN surgical oncologist. And so we do a lot of her cases. And then Dr. Wong is one of our radiation oncologists, and he is a big Caris fan because he treats mainly CNS tumors. And these are examples of some of the things that were found. And so everybody mix and matches. But again, remember, for every Caris test, there's a Signatera and there's a Tempus and there's something else. Okay. Oh. I'm not moving now. Okay. Oh, there. And this is a case just to remind everybody, we used to do a lot of Precise with Myriad. We don't use it so much anymore. But in addition to the genomic profiling, I show you this here, and everybody's doing this now. So Empower does it with Natera, Tempus does it with Ambry, Invitae, we do germline with them quite a bit. Having said that, we get notifications from everybody that this patient meets a criteria for germline testing. And if we haven't already tested them, we flip around and we test them. And this is a guy who came in with colorectal cancer, but then lo and behold, he winds up having a RET mutation for medullary thyroid. So now he's under completely different guidelines than he was what he came in for, which is a good thing that we've identified something in someone that we can actually prevent. Okay, am I running over? I am. Okay, let me just finish real quickly. Sorry. So I have to give Menarini a shout out because our new thing, my patient, my 53-year-old neighbor that we can't figure out what's going on. So we have gotten into doing circulating tumor cells. So circulating tumor cells, not circulating tumor DNA, not cell-free DNA, but whole intact cells. And this is from Cell Search, and I used to do a lot of this. So what we're doing, and this is relevant to HER2, it's also relevant to cancer in general. We have HER2 conversion, and you can have HER2 conversion in a patient that crosses over. So we're doing this more. So my 53-year-old woman that we can't explain why she keeps having rising signals, we're gonna do a circulating tumor cell on her to see, are there any tumor cells circulating in her periphery? The original Garden Reveal test I showed you with the lung cancer that keeps going high. Also with her, we're gonna do, our radiation oncologist has ordered a circulating tumor cell on her. And this is kind of a big deal. We did this years ago, it went away, and now it's back. And at the end, there's a lot of different things and all, but the conversion rate is high. So you can't be negative. In the old days, it used to be once you were HER2 positive, you were always HER2 positive. That's not the case. So anyway, I basically am a geneticist by training. I love what I do. I love what I do now. It's a little bit different. I don't glove up and gown up in the lab anymore, but I do educate patients. And part of my whole thing is to educate patients and let them understand, whether it be circulating tumor DNA, whether it be Garden Reveal, whether it be Caris, whether it be Tempus, whether it be Invitae, Myriad, Hologics. We do a lot of breast cancer. And I mean, we're all over the place. So anyway, thank you guys for listening. I'm sorry I ran over a little bit.